Hi everybody, thank you so much for joining this afternoon's webinar in collaboration with the University of Southampton. We've got a fantastic panel of speakers on the call for you this afternoon. And we're going to be exploring all of the different options within biological sciences, the degrees you can study and some of the different careers you can go into afterwards too. Whilst we're waiting for everybody to make their way out of the waiting room into the main session, I just want to point out the Q&A function at the bottom of the screen. If you've got questions during today's session, please do pop them into that Q&A, and then we'll have a nice 10 minutes at the end where we can go through and get as many questions as we can answered. But I won't take up any more of the time on today's session, and I'll hand over to Neil to do an introduction to the University of Southampton and what we'll be covering today.
Hello, my name's Neil Gostling. I'm an Associate Professor in the School of Biological Sciences at the University of Southampton. My area of study is evolution and paleobiology. So I work on all sorts of different things. My background is looking at genes that pattern the nervous system in chordates, that's the group of animals that we sit in, but that pattern the segments in flies. And so I was trying to work out what was going on there, and it was a lot more surprising than we thought. It turns out flies and spiders are special, and humans are actually quite primitive for gene function. I want to give you a little bit of an overview of what the School of Biological Sciences looks like, and then I'm going to take you through the reason why I study zoology, why I'm interested in this, why this is an exciting area of research, and how students actually in our school can get involved with it. So our lecturers come from have a really diverse research background. We have 7 research themes: molecular and cell biology, neurosciences, plants and food security, developmental biology, systems biology, microbiology, and ecology and evolution. And I wouldn't be surprised if most of those made no sense to you whatsoever. The whole point of this is to try and explain and show you the huge breadth and diversity of areas and disciplines within biology. We all know what biology is. We all think we know what biology is, but actually it goes from everything from the tiniest molecules all the way up to whole organisms and ecosystems. Anything to do with living animals, living plants, life, from the molecular to the ecosystem scale is covered by people working. I'm going to talk to you a little bit about the work that I do. The theme that I am in is in ecology and evolution. So I'm very lucky. My first degree was botany and zoology. I then went on and did a PhD in evolution and developmental biology, having worked with embryos of lots of different worms and mollusks and squatty little animals that no one cares about. When you say embryos, people often think of like the little baby on the Pampers advert, and they go, oh, isn't that like, that's not an embryo, that's a baby. An embryo is everything from a fertilized cell, fertilized egg, through all of the developmental stages, all up to the point at which that little animal hatches out and does the rest of its thing. And embryology is understanding how you get from that single fertilized egg all the way through to a fully functioning, free-living individual. I worked on lots of those embryos. I then went into looking at fossil embryos, so embryos of jellyfish and arthropods and little worms, from 500 million years ago. So some of the very earliest animals that we have in the fossil record, I was working on those to try and understand how patterns of development had evolved, because it's during development that evolution plays this exciting part, and that changes can happen, and that give rise to different species and all our understanding of evolution as we see it. However, the big things, the exciting things people think of when you think of evolution and paleobiology, we automatically jump to dinosaurs. And I'm really lucky because I work on dinosaurs and pliosaurs, which aren't dinosaurs. Dinosaurs live on the land. If you live in the water, you're an ichthyosaur or a pliosaur or a plesiosaur. If you're flying through the air without feathers, then you're a pterosaur. Dinosaurs are a very specific group, and I work on these wonderful, wonderful animals. I use CT scanning to get inside these fossils. So this here, we have the Etches collection pliosaur. It's the largest and most complete skull of a pliosaur, a Mesozoic marine reptile from the Jurassic. It's the largest and most complete skull that we've ever found, and it's absolutely wonderful. And in order to understand it better, you can see up in the top right here, we have its snout strapped to a very weird contraption inside our CT scanner, and we were able to penetrate the whole fossil, and we were able to find nerves and blood vessels and see how teeth were replaced and help to really understand this animal a lot better. I work on dinosaurs. Here are two specimens. We have in the foreground, we have Ceratopsuchops, which means horned crocodile face. It's got a horn and it's got a long snout, a bit like a crocodile. And over in the back there, hunched over, and roaring over a dead iguanodon, is Ripara veneta, which means riverbank hunter. And again, these are spinous from a very exciting part of the world. You don't necessarily think it's an exciting part of the world because it's the Isle of Wight, and we sort of think, oh, it's where people go on holiday. The Isle of Wight's wonderful. It's the best place in Europe to find dinosaurs. It's eroding at between 1 and 2 meters a year on the south coast, which is wonderful because then fossils, it's not great if you own a house at the cliff. But if you are interested in things falling out onto the beach, dinosaur fossils, great, brilliant, wonderful. They're there. And the fossils fall out. They're collected by people, taken to the local museum. The local museum get in touch with me and say, we've got some new dinosaurs. Would you like to have a look? And I say, yes. And we've been very lucky. We've worked on these 2 new spinosaurs. Dinosaurs are weird. Dinosaurs, I've just said, are terrestrial animals. They live on land. And these are clearly on land, but they're also associated with water. And these dinosaurs eat fish. Fish is not a normal part of a diet of dinosaurs. Dinosaurs are either eating plants or eat other. Spinosaurs do eat iguanodons. We've found a specimen with the bones of a dinosaur inside it, slightly acid etched, suggesting that they've been partially digested. But they've got these long snouts with these conical teeth, just like crocodiles. And along with that, those bones of a dinosaur that were found, acid-etched fish scale. It's not just a fish that's died and floated in the water where the dinosaur died and it's landed in roughly the stomach region. No, this is a fish that was eaten, has been digested, and then everything's died, and we've managed to find it inside its belly. So this is a weird animal. We've also found another spinosaur. We didn't name this one because we only had 5 bones, but the bones we had showed that it is a massive, massive predator. It would have been between 11 and 13 meters long. That makes it the largest predatory dinosaur that's ever been discovered in Europe. Again, on the south coast of the Isle of Wight. At the time, 125 million years ago, give or take, there would have been a large river system flowing out from what's now Cornwall, opening up into a large floodplain with mud flats, and dinosaurs would have been, we have this wonderful term we've come up called dinoturbation. Basically means dinosaurs have been walking through the mud, leaving their footprint, leaving behind their fossils, leaving behind the evidence of them being there. And we find this all on the south coast of the Isle of Wight. It's absolutely wonderful. Here we had a look at Ceratopsuchops, that was the first one that I showed you in the foreground, and tried to work out how it was eating fish. What is it? We're very lucky with Ceratopsuchops and Ripara veneta. We have 3-dimensionally preserved brain cases, the skulls, the bit of the skull that the brain is in, which means we can take an internal mold of what it is. We can look at it, and up in the top left-hand corner there, you can see the mold of the brain, the shape of the brain. The yellow shows you the major cranial nerves. The pink is the semicircular canals. We've got these you can work out exactly how sensitive the face is. You can see the bit projecting up towards the sort of top right, roughly pointing towards 2 o'clock. That is the olfactory bulb, so we can tell how well it could smell. We've got 2 little yellow, I don't know if you can see the arrow, there's 2 little dots here. Those are the bases of the optic nerve. We've got the trigeminal nerve, which is the nerve, if you touch your face, you can feel your fingers dancing over your face. That's because you have a trigeminal nerve branching up into the top and into the bottom and into another part of your skull as well, which allow you to have sort of facial sensitivity. Semicircular canals, how well could it balance? We've got this projection here as well called the flocculus of the eye. It works with the eyes and allows the brain, the eye system to work with the semicircular canals for balance and work out, keep your eyes fixed. We can work out how well the whole neurosensory system of this animal works, so that we can try to work out how it could catch fish. And it turns out that it could smell just as well as any other dinosaur, no better. It has no better facial sensitivity than any other dinosaur. Its visual acuity, how it can lock up, is no better than any other dinosaur. The thing that has changed is the shape of its head, the shape of its teeth, and the fact that it has the same shaped teeth and the same shaped face as a dolphin, as a crocodile, as an alligator gar, which isn't an alligator, it's a type of fish that catches other fish. These are adaptations to allow animals to catch fish. So T-Rex doesn't normally eat fish, but actually it has the neuroanatomy, it has the brain power. If it wanted to eat fish, it probably could, but it couldn't catch them because its jaws are too big and the wrong shape. Ceratopsicops can catch fish because its jaws are adapted. So it's mechanical changes, not neurosensory changes. So I'm very lucky to have worked on several different spinosaurs and to be able to have a look and see what these animals are doing. Birds are dinosaurs. Understanding the way that birds develop is really important. Understanding the shape of populations and the population structure in the Mesozoic is really important at the time of early birds. And there are features of bones which allow you to determine if a bird is male or female. At certain times of a female bird's life, she's laying eggs. While she's laying eggs and leading up to, she produces a bone tissue called medullary bone. This is a sacrificial calcium source, and it's the region that the bird can get calcium to make its eggshell. If you've got medullary bone, you are a female. If you haven't got medullary bone, you are either a female not laying eggs or a male. But you can't, but you know, if you have the medullary bone, you have to be female. So it means that we can have a look and we can start to work out, maybe from fossils, if it's a male or a female. There is a problem because there's a disease, mostly in chickens. It's called avian osteopetrosis. Osteoporosis is the loss of bone structure, the loss of bone mass. Osteopetrosis is the overgrowth of bone mass. And sometimes you get in chickens, you get a viral infection that causes bone overgrowth, and we get this structure that looks very similar to medullary bone. So we have to work out if they're different. And luckily, I have a PhD student who I'm supervising. She's absolutely brilliant. And she has shown that there are differences in the porosity, in the connectivity between holes, and the way that the structure is, the thickness of the bone, the number and shape and orientation of osteocytes, the cells which secrete the minerals that make bone. They are different. So we can tell the difference between osteopetrosis and medullary bone, and so we can use this as a marker in the fossil record to help us understand population structure. I've also worked on Archaeopteryx, the first bird, 150 billion years old. How could Archaeopteryx have got off of the ground? It couldn't flap very well, doesn't need to flap. Birds don't get into the air by flapping their wings. Birds get into the air by squatting down and jumping. It is the hind limbs that get them airborne and their wings take over. And what we worked out that is if an Archaeopteryx takes one jump, flaps, another jump, and then flaps, that gives it a 400-gram Archaeopteryx enough force, enough punch to get to 7 meters per second and enter level flight. So we've demonstrated theoretically how Archaeopteryx could have got off the ground. And this is important for understanding how the transition from dinosaurs into modern birds. Finally, I've been working on the dodo, which is an absolutely wonderful animal. The dodo is the first species where we recorded its presence in about 1598 and then recorded that it had completely disappeared in 1662. No more sightings after 1662. In fact, and almost certainly extinct by 1681, which shows you that in less than 100 years, humans had got to Mauritius. They didn't eat it to death. We often have this idea that Dutch sailors got there and thought, well, that looks like a big fat tasty bird, let's eat it. They didn't do that. What happened? Well, they did try it, but it was fatty and greasy and disgusting, and unless you cooked it really well, it was too chewy and horrible. But the cats and the rats, they weren't so fussy. The pigs weren't so fussy, they ate the eggs, and goats just trampled on everything. Within less than 100 years, the dodo was gone. And it was also gone before we had a good understanding of taxonomy and nomenclature. And so how we have classified it has changed a number of times over the last 400 years. So we've spent an awful lot of time going through and trying to work out exactly how it's related to everything. It's a big fat pigeon. But it turns out it's not actually that fat either. We're doing a biomechanical study at the moment to see how it would have moved, how it would have walked around, because it was a really important part of the habitat of what is Mauritius. It was really important for maintaining the and growth of the forest. There's a tree called the Calvaria tree there are no trees naturally germinated for the last 400 years. That basically the fruit was eaten by the dodo, passed through the dodo, was deposited in a healthy little deposit of fertilizer, and then germinated. No dodos, no Calvaria trees. These were important animals in the ecosystem, and so understanding it better might help us to mitigate some of the changes which have happened now. Very last thing, I work on fossil mammals too, and if you come to Southampton, I'll tell you all about that. One of the things I would say is all of those projects at some point have involved students, and where publications have resulted, those students have been named on the papers as authors and have scientific publications to their names. I shan't touch on this now, but if anyone is interested in what we do and they want to know a little bit more about life after graduation, you're very welcome to contact me. I'll pass over to Elisa now, who will tell you about what she does. I shall just stop sharing and let her share in my place.
Yeah, thank you, Neil. Okay, I'm sharing my screen. Yeah, there you go. No. There, from the start. So hi, everyone. It's my pleasure to be here, talk to you about how I became a glycobiologist. That's a big word, and it has nothing to do with what Neil just said. So I will have to explain what it is, and I'm going to go through that via a journey through what complex carbohydrates are. So, well, what are glycans? Glycans, and where the word glycobiology comes from, are sugars. So I'm a sugar scientist. And all of you have some experience with sugar. And some of you might love sugar, like myself, but not everybody knows that sugars are actually everywhere. So sugars are the most abundant molecule or polymer in nature, because they're present not only in our beloved sugar that we put in tea or in sweets, ice cream, but in pretty much everything that surrounds us. There's soluble sugars, which are the ones that I just explained, but there are non-soluble sugars. For example, the shell of crustaceans like a crab that I'm showing here, or insects, are made of sugars. So sugars are enormous variability, and there are molecules at the molecular level. They're just like this connection of units called monosaccharides, or glucose in this case that I'm showing you here, but they're also branched. So they kind of look like tree. And they constitute the largest component of the biomass. So there's a lot of richness and interest in sugars and the sugar biology in every aspect of biology and health and disease. So, but how I became interested in that, not just through food, and in particular, it's like in the area of glycobiology, not through food or my love for sugars or sweet or ice cream in particular, but through a very winding road. So if any of you has not a clear path of what you want to do when you grow up, but you know that you like some things now, stick with those things. And the road is very fun to follow. So I'll show you my road. And how I became a glycobiologist, actually, upon reflection, started in an odd place. Started in 1982, which is like medieval times for your perspective. But I was very, very young at that time, and I was absolutely in love with video games. So video games that in 1982 were really basic. I don't know if any of you knows that Snake kind of game, retro game. So they were beyond retro games. And my dad decided not to give me a game console, but that I should have been learning how to do programming. So he enrolled me in a programming course, which taught where I was taught basic programming language, and it gave me for my birthday a ZX Spectrum, which is one of the first home computers, and I was delighted with it and had so much fun. And I learned how to program my own video games. So that was the start of it all. And you'll understand why this is the start of it all in a second. So this is the twisted road that brought me to where I am now. So like, and you'd be surprised that in secondary school, which is the studies that you're doing now, I actually did anything but science. Like, so I actually was studying classics, and I studied Latin and Greek and how to translate ancient texts. So, and that was very, very useful. And I loved it, and I was quite good at it. But I decided that that was not what I really, really loved. I love science because I was curious. So I decided to go to chemistry in university, and I did an undergraduate degree in chemistry, followed by a PhD in quantum mechanics. So like, which is between chemistry and physics, because I really wanted to understand how things worked at the basic atomistic level, how nature works. It was my main interest. So then I decided that like the atomistic level was a bit too small, and I decided to study biophysics. That brought me to the scale of large proteins and large molecules and how they're involved in health and disease. And I worked in a hospital in Canada, in Toronto, Canada, to do that, which is a magnificent experience. And then I actually encountered glycobiology for the first time at that hospital. And I thought it was like a completely mysterious universe because the sugars were unseen. It's something that nobody could see for different reasons, because they're very flexible, they're very small, like, and they're very fascinating, and they're not determined by DNA, so they're not encoded in your genome. So it's like it's such a mystery involved with their importance in health and disease, with the connection through the hospital, got me into thinking I could combine computing, which I loved since 1982, with glycobiology to become a computational glycobiologist, which is what I am now. Well, as I started as a lecturer, like having my own group many years ago, and now as an associate professor at the University of Southampton. So what is computational biology? Well, I actually grew up like in terms of the computers that I use. Like, so I started with the ZX Spectrum, and now I'm using rooms full of computers like called high performance computing. So, and we are able to do simulations of molecules, whole molecules in models of the environment where these molecules are found, which could be on a cell or in water, and their interaction with other molecules, and where the main focus of my attention is the sugars that grow on those molecules. Some examples of the stuff that I study is viral glycobiology. You might recognize the name, the SARS-CoV-2 spike, which is like the protein that actually mediates the infections of the SARS-CoV-2 virus that actually caused COVID. And then one of the pieces, the arsenal, like the pathological arsenal of Neisseria meningitidis, that, as the name suggests, is the trigger for meningitis. And also in completely normal, or like structures that exist in completely normal systems or beings or humans in this case, and how the sugars in this case is a heparan sulfate type of sugar interacts with our own biological molecules that make us alive and makes us function. So I can see all these little sugars that you can see here colored in blue for the SARS-CoV-2 virus, in different little colors, like for the peeling that you see here and here, like in where you can see the atoms, so in cyan, and in green for the TSR domain. So like why the importance of sugars, and why I study sugars in biology, in the context of biology. But this shows like a cell. In this case, is a bacterial cells from a Bacillus subtilis, and you see that this cell is furry. It looks furry. Like you can see this kind of like fur that stems out of the cell wall, and this is actually like a very, very visible in the context of bacteria. Because they have very, very long carbohydrates, but like all our cells are furry to different degrees. So I studied this fur, like how carbohydrate, what's the carbohydrate function on the cell wall. And this is a representation, graphical representation of what this fur can look like, so where the carbohydrates are. But what I like to think carbohydrates work as is like little hands sticking out of the cell surface and interacting with whatever comes towards the cell, which could be its environment, other molecules, such as lectins, antibodies, or carbohydrate active enzymes. So anything that surrounds it, other cells, but its hands also facilitate and mold the protein structure, keeping it together and keeping it compact and making its 3D structure functional. But also a handshake is the first port of entry of toxins, bacteria, and viruses. And this handshake with carbohydrates is how you get the flu, for example, which is what I am really interested in, understanding all this different handshake at the molecular atomistic level detail. So what I worked on for a long time, for a long time since it came out, is the SARS-CoV-2 virus. So you can see here, actually, the SARS-CoV-2 virus, and you can see those spikes that I mentioned earlier, like, so the proteins that actually mediate infection. And they look, they're called spikes because they literally look like spikes coming out of the cell envelope. And they can be, they're very similar to this, let's say, macroscopic model of the SARS-CoV-2 virus that I found in the park while walking, doing lockdown, which is a shell from a horse chestnut tree. You can see the spike. So the spike of all these viruses, they're quite similar in terms of their preferred host is actually humans, is that they cover the spikes or the proteins that mediates fusion with the host cell. It's covered with sugars. And the viruses use the sugar as a cloak. So the sugars hide the identity of the virus from the immune system. So the virus can go through your body without being detected by the immune system, especially when your immune system is not trained by a vaccine. So we cannot see this cloak because, as I said, this fur moves very, very fast, as in this case of somebody taking a picture of a very busy New York street. You have people moving and like what you see is only a blur. That happens the same way when you have a subject like a sugar, that you try to take a picture through X-ray or electron microscopy, and you have just like that you cannot focus on it because they move too fast. So I use high performance computing, the famous room full of computers, to recalculate and bring back the sugar, the identity of the sugars. So this is a spike from the SARS-CoV-2 virus that it doesn't have sugars. So you can see through cryo-electron microscopy. So it's a camera for molecules. And it doesn't look like anything like what the spike actually is in reality. It looks like a shaved version of it. This is the spike that we reconstructed through millions of hours of computing through those machines. We used actually the 8th most powerful computer in the world to rebuild this spike. And this is what your immune system sees. So the bottom of the goal, like let's say, like the aim is not to build video games like the same aim that I had when I was 9, but is actually to understand how like the sugars can protect and camouflage the virus by reconstructing the actual virus, what it looks like, and to understand how to design small molecule for as drugs, therapeutic antibodies, or in this case, vaccines. So for those of you that had a Moderna or Pfizer vaccine, I had 9 of them. So I can tell you that I had loads of this spikes built on my cells. This is what that vaccine looks like, which is almost identical to the actual virus spike that you have if you get COVID. So how do I become a glycobiologist? Well, there's not really a recipe for it. Like, so the more I reflected about it, it's like a very, very winding and fun road. The common denominator are very simple. I really like to study. Nobody told me to study, you have to study, you have to sit down and do this. I actually liked it a lot. Not everything, I had my favorite things, but I really did like to study because I was always being curious about how things work. So like I really like to look for information and think about it. I actually never made long career plans. So if some of you are lost or like, I don't know what I'm going to do when I'm 50, well, no worries. I never knew what I was going to do next year. So now I think I know, but when I grew up, I actually didn't. I really followed my interest and tried to combine them. That's why I became a computational glycobiologist, because I always liked computing. So I also enjoyed traveling. I wasn't really a rooted person. So, and I like to meet and work with many people and discuss things together and try to find answers. So, and I try to have fun at each stage of my career. Still having fun is not worth doing things if you don't have fun while you're doing them. So, especially in science and research. But one thing that actually resonated always with me, and I think it's just like why I became a glycobiologist, is because I didn't ever listen to anyone telling me it's too difficult, you'll never succeed. This is sugars, nobody cares about sugars. I still hear that today, and nothing is gonna deter me from my goal of continuing my studies. And I understand how sugars modulate biology and response. So thank you very much. Hope it was useful to you.
Thank you both. I think a really lovely message coming out of both of the different sessions, however different they might be in terms of topics, is the fact that you can turn a passion that you have into a career, and there are different degree pathways out there and different career opportunities to really let you continue to study something that's really exciting and that you enjoy. And like you were saying, Elisa, in terms of being able to sit down and study and nobody's forcing you to do it because you're enjoying the subjects that you're doing. And that's really the opportunity that you have at university, is to pick that one degree that really stands out to you and is something you're going to enjoy studying in great detail for 3 years and potentially even further if you do take it on to pursue as a further degree course, maybe a master's or a PhD even. And what's really come through from the 2 sessions we've had today is how different and diverse degrees and careers in the biological sciences are. We have had some questions already start to come through, so we'll start working through some of these, but students do continue to ask questions in this sort of last 10 minutes of today. The first one came through, Neil, whilst we were chatting through your first portion of today, and it's a student with a particular interest in dinosaurs, and they were wondering about the images that you were sharing and said, are those depictions of dinosaurs accurate, or did those types of dinosaurs not have feathers?
That's an excellent question. Okay, so these are accurate representations. We're talking about animals here which are between 9 and 13 meters long. If you are a small, okay, so we know that pterosaurs and all dinosaurs primitively have feathers. However, if you get big, having feathers is a disadvantage. Elephants are big and they have big flappy ears to help them lose heat. If you are an animal the size of an elephant or the size of a theropod dinosaur, such as Ceratopsuchops at 9 meters, Titanosaur with a 15-meter neck and a body which is 35 to 40 meters long. If you are covered in feathers, you will cook yourself, and that is not a good argument for survival. So as a chick, because eggs are this size, coming out of the egg, running around being covered in feathers would be advantageous, but as you get bigger, no. So T-Rex wouldn't have had feathers. It might have had some quills, it might have had something for display, but an adult T-Rex, definitely no feathers. Adult Ceratopsuchops, no feathers. Chicks, yes.
There we go, fantastic. I'm sure that student, yeah, that's definitely answered their question. I'm learning lots today too. That's what I love about these webinars, is I get a nice taster of lots of different subject areas. One student as well, Neil, is probably sat there and thinking, this is exactly what I'm interested in. I didn't know there were careers in this. They've simply asked, what would the job of Dr. Neil be called?
The job of Dr. Neil would be paleobiologist. There's 2 routes you can go to getting into paleontology. Broadly speaking, I'm a paleobiologist. I'm not interested in the geology per se. I'm not interested in the structure of the rocks. I'm interested in the animals that happen to have been dead for millions of years. So I'm doing biology on things which are ancient and gone. I'm a paleobiologist. So you can get into that by coming and doing biology or zoology and then doing master's degrees in paleobiology, and then a PhD.
Fantastic. We will come on to you in a second, Elisa, but you keep hinting, Neil, to different questions that are coming through. And you mentioned zoology then, and there is a student that has asked, should they start with a broad undergraduate degree in biology or go straight into zoology?
Why mess around with a broad degree? Go straight into no. Look, if you're interested in how animals work and how they work at all scales, from a cellular level through to the ecosystem, and you want to study animals and understand them, a degree in zoology is not going to allow you to work in a zoo. You need all sorts of qualifications in zoo work to do that. Zoology is the study of animals. If you are interested in them from a cellular level to an ecosystem level, come and do zoology. It's what I did as an undergraduate. I did botany and zoology. It's a brilliant degree. It's absolutely relevant for all sorts of jobs and everything, not just staying in any degree you do is going to give you lots of skills to go off and do anything you want to do. If you're interested in animals, come and do zoology. It's even relevant to doing something like Elisa does, working on COVID. COVID came from bats. Understanding bats allows you to so it's all there. Zoology is really important. So yeah, if you want to do zoology, come and do zoology.
Fantastic. Yeah, and I think we'll probably touch on this multiple times throughout, but open days are that fantastic opportunity if you're stuck between different degrees to go and sit in some different talks and see the different modules and really find which one gets you super excited and you can
go to all the open days you can possibly go to. Go and find out what we're doing. Not just Southampton, go to everywhere and then decide Southampton's best. But come and find out what we're doing, and we'd love to see you.
Fantastic. Thank you, Neil. Moving on, Elisa, to looking at more your side of the biological sciences. A student has said that they're interested in medicine, but they also want to apply to other sort of biomedical, biochemical programs. Obviously, what you're studying at the moment is quite specific, looking at glycobiology. Is that too niche at undergraduate? Where should they start when they're looking at undergraduate courses to take them potentially on a similar path?
Oh, like absolutely, it's not too niche at all. Like it gives you like a good perspective, and if you do not like, there's not a degree in glycobiology. Actually, it's somebody asked Caroline Bertozzi, who won the Nobel Prize like 2 years ago for chemistry, and she's a pioneer in glycobiology. She's one of the people that invented glycobiology, and she said, like, what is the degree that you had to? What is the subject that you had to teach yourself? And she said glycobiology. So it's kind of like it's particular type of study that takes from chemistry, from biology, from biomedical sciences, from biochemistry. So and you have to do all or some of those before you become so specialized. So like starting with biochemistry and biomedical sciences or pharmacology, for example, it's a good strategy to find out what you really like. On neuroscience, like I don't want to take anybody out, like it's to find what you really like. In chemistry, which is the degree that I chose, there was a bunch of stuff that I really didn't like. I decided like I am not going to be an organic chemist. So, but it was useful for me to study it because I mean, it's a huge part of glycobiology is organic chemistry, sadly, but it is, and I'm glad I know it.
Yeah, and I think it's one of those, isn't it, that as important as thinking ahead to career is when choosing your undergraduate degree, when you're doing that study for 3 years, you will find things that you didn't know existed when you first began that degree,
Exactly.
and it will take you off on lots of different pathways. Yeah. One student that's considering biomedical science, which we've spoken about a little bit as well, is wondering, will they struggle with that course if they're not currently studying chemistry at A level?
Well, James can tell me exactly, but I think it's required.
So you don't, to apply, you don't need chemistry. But if you were to come to university, there's a first year chemistry module. And that chemistry module is really to bring everybody up to the same level. So if you don't have chemistry before you come to university, in the case of Southampton, then when you arrive, we have modules in place that will give you the support to develop those skills in chemistry that you would then be able to use as you progress through your degree in biomedical sciences.
Yeah.
Fantastic. Thank you very much. And we're talking about lots of different degree pathways here and different programs, because there are so many for students interested in the biological sciences. One student has asked, how many years of study do these types of degrees normally require?
Oh, like how many years of study? In general, for me, I'm still studying. So forever. And that's the fun of it. And I'm sure like Neil would agree with me. It's like that's most of the fun that I have is when I am left alone and I can study. Like it's that I don't have to deal with other things. It just like, reflect and study and look at things. And so how much studying is required, it depends on what you want to do. Like some of us has never haven't finished yet, like, but like a BSc could be enough. Like if you reach the amount that you decide this is enough, this is what I want to do now.
So this is very true. I completely agree. I mean, the question about how long study is there for these degrees, 3 years, 4 years if you do the masters. If you now, okay, I'm going to let you into a secret. Okay, all of us sitting at the top that you can see in this row, or however we are in your computer screen, we're a bit weird. We're a bit weird because we are still doing what we did when we were undergraduates. Most of my friends at university are off doing other things, and they're making money as bankers and all sorts of other things. But they're not having fun. They're not having a nice time. We are still studying. We are still working out how the world works. How we're asking questions every day. We get to work with students, and it's always fun to work with people who are interested in what you're doing. It allows us to explore and think about things in different ways. If a student comes and asks a question, it might be the first time. Look, people from with different backgrounds, people with different ideas, ask questions differently, and asking questions differently makes you think differently. And so we are absolutely still learning, and it's the best job I could imagine. Yeah, I mean, obviously the problem is that some of you will come to university, you'll be really, really good, you'll work really, really hard, and I don't want you to stay. I want you to go off, do a job somewhere else, because I know that you'll be better than I am. And if I encourage you too much, you'll be chasing me out the door, and I like my job too much. I know, in all seriousness, it's the best thing. If we can get people here who are enthusiastic, who are excited, the sky is the limit for where it can go. And honestly, every single day you will be learning more. Every single day you will be studying things. Every single day you will be working in a subject that you absolutely love. That's the whole point of university. Whatever job you go off and do, find a degree that you love, that you're really passionate about, that you're really excited about. That will give you all the transferable skills that you need to go off and do any other job that you might be thinking of. But why wouldn't you get those transferable skills in something that you enjoy? If you like working on animals, come and do zoology. We'll teach you statistics. We'll do all those things which are, but we'll teach you them with the flavour of animals. We'll teach you with the flavour of plant sciences. Someone asked a question about what they should do if they want to do plant sciences. Come and do biology. You can do all sorts of plant sciences modules. It's a great opportunity. And honestly, find that thing that you love and spend 3 years, embed yourself in it. Take every single opportunity you can, and you'll have a wonderful time.
True.
I think that is very great advice to sort of round off today's session. I know we're going into the final minute now of today's webinar. I suppose sort of to round off everything that we've spoken about, because we've spoken through quite a lot today, what would you say for students that are preparing to study at university or thinking about writing applications? What would be one top tip for what they can be doing right now to put themselves in a great position for either applying to university or deciding on which subject area they can do? So just one top tip for the students on the call. Elisa, I will put you on the spot first. What would your top tip be?
Like what I have done, I can only tell like what I've done, like as an experience, lived experience. I actually, coming from a background, because like, I mean, as I said, I like in secondary school, I did the classics. So, and I really, like, I reflected and was just like, do I want to continue this? And I was actually good at it. So I could have, like, I was encouraged to do that, but my heart wasn't in it. And somebody asked about chemistry. I was disadvantaged at the beginning choosing chemistry because having studied classics, my science background was very, very poor. compared to some of my colleagues, but I studied hard, and like I was brought back, as James said, to the same level everybody else. Actually was studying harder because I knew I didn't know things. So, and I discovered that that is what I like. This is what I was, that what was my calling. So go for what you think you like. If you're not sure, it doesn't matter. There's time to change, but just follow your instinct and have fun. That's the most important thing. So like choose something, as Neil said, that drives your passion. That's so important because there's a lot of work to do. So you have to have fun while you do it.
Great, yeah, I think that's a wonderful message that's sort of been threaded throughout today. And Neil, we're going to go for a quick 10-15 seconds because of the time. What would your top tip be?
My top tip would be find out that thing that you're passionate about that you really, really love, okay? There is a question, and I'm going to answer it, and I'm going to answer it in the way that's absolutely related to this. Find that thing that you're passionate about. You've got 3 years. You've got to get the best degree that you can get. If you get a third and you get or first, you're paying back the same amount of fees. So find that thing that you love and get the best degree you possibly can. Is zoology good for job prospects? Absolutely. It is a degree that will get you a graduate level qualification. All degrees will get you that same, in science, for example, will get you that same baseline of qualification and allow you to go into any graduate job related to sciences and biology or whatever it is you happen to be in. It's the transferable skills that you'll get. Find the thing you're passionate about. Come and study that, because that will help you. If you're interested in it, if you love it, that will be the thing that enables you to get up on a 9 o'clock on a Monday when you've had a heavy weekend doing whatever students do, get you into class, get you into the lectures, because if you come to lectures, you get 10% higher on average mark than if you don't come to lectures. So find that thing you love, do it, and have a brilliant time.
Great. You're both making me want to go back and go to university again, have fun for 3 years, study something you're really excited about and really enjoy. So thank you all so much for your time this afternoon. I know it's been an incredibly beneficial session for the students, which has been shown by the questions that are still continuing to go through. We will be sharing the recording from today, along with information of how you can continue researching with the University of Southampton, the degrees they've got on offer, and going along and visiting at an open day too, where you'll be able to get much more detail about the courses on offer and continue to ask those questions that you have. But we'll round it off there for today. A final thank you both to the students that have joined us and also to our fantastic speakers. Hopefully we'll see you on another Unifrog webinar soon. Thanks everybody.
Thank you.